First of all, I really want to thank to the organizer, Dr. Zohair and his team to inviting me, and of course the university and sponsors to sponsoring him to organize this meeting. Really great meeting. Regarding my work, Avian Influenza, I would say that currently in the world, the top poultry disease is threatening I would say the poultry industry is avian influenza. The reason I would say that what the topic is saying that challenges. The challenges are many. One of the major challenge is because of avian influenza infection in Europe and USA in North America, and of course, we here in Pakistan and in countries in South Asia are using vaccines. We are a little bit protected, but the risk always remains. And the challenge is to the public that in Europe, the prices of food, particularly for meat and egg, is raised about 40%. And in USA, it used to be in $1 you can buy 12 eggs, and now 1 egg per dollar. So this is all comes to the public. And that's the authorities, including the researchers and the veterinarian and all, are trying to control avian influenza from poultry, but the avian influenza is threatening and a huge risk because this virus is in wild birds. And the second is what is the control strategy? The control strategy used to be just a biosecurity in USA, in Europe, and now because the virus is highly prevalent or circulating in wild bird, and if even you have increased your biosecurity highest in your farm, still the virus one way or another leap into it and causing outbreak. And virtually every week in UK we are experiencing a very high biosecurity form, or this biosecurity is being breached, and a lot of outbreak or being reported in the UK. And last 3 years, around 20 to 30% farmers lost their business because once their farm is gone, they didn't bother to actually restock it. So therefore, the threat of avian influenza is huge. And the Poultry Institute where I work, our aim is that to predict, to do research, to predict, to do surveillance, and see how we can improve the control against these viruses. So at Poultry, as you can see, my group is avian influenza and Newcastle disease, but similarly there are other group leaders. Is around 27, 28 group leaders working on different animal diseases, and some of these diseases, of course, directly transfer to humans. So, in terms of avian influenza, as you can see, there are so many subtypes, and the subtypes, the gene of these viruses are segmented. So they can change each other. For example, if you have H1, it can be H1N1, H1 and 2, and so on, based on its surface protein, which we call it neuraminidase and hemagglutinin. And these two proteins are antigenic. Based on antigenicity, these viruses are being exchanged their genes, and it becomes like H5N1 and so on. And last few years, these viruses have been isolated in different subtypes, and some of these subtypes, all of them isolated from wild birds, some of them in poultry. And now the biggest risk also to the cow, because in USA, there is so many outbreaks, more than 140 farms in 16 states have been affected, and each farm may have a 90,000 or 200,000 cows, and as you can see, in one farm, if you cull 104, I mean 90,000 or 100,000 cow, what's the cost of that is? So the main reason is we are studying how these viruses gain fitness to infect poultry, and from poultry or the wild bird to other animals, and then of course the risk to human. So for poultry, so far there are few subtypes, H5, H7, H9, and H6 is known to can establish in poultry, and then it keep on changing, and we called it evolution. And this evolution changed its virulence, its transmission, its pathogenicity, and of course its ability to cross species barrier from, for example, from poultry. The virus is poultry virus, but if you give it exposed to human, then the human can be infected. And then some subtypes have more ability, and some subtypes have less ability, or some variants generated which pose more risk. So our aim is to risk assess those which are emerging viruses, and which I describe you later. So at least in Europe or in US only, which is a reported outbreaks, more than 300 million birds have been killed or died. And similarly, it is reported that over 300,000 birds or 60 different wild bird species have been died or affected. Similarly, mammalians, dairy cow in USA, and some forms like minx form and sea lion sparrow. So the wild dice is also directly affected, and certainly last year, even in human, more than 70. Luckily, this virus is less severe, cause disease, but still it causes infection to human. Again, in Pakistan, H9N2 is endemic. It is always causing threat to poultry in Pakistan. And we here do vaccination, but in some countries like in Morocco, before vaccination they start, they reported that their bird, when they have H9, up to 70% of their laying hens or breeders stop laying, so 70% reduction in egg production, and they see the mortality up to 60% of the broilers. And similarly, of course, if the broiler reduces feed conversion, of course, they will not get the weight gain, and then a simple respiratory signs of respiratory infections, which are RAWs and lacrimation and so on. And one of colleagues here, sitting even here, he gave this video, which not sure I can run, but anyway, the time is not. But these are the clinical signs you may see, and maybe some veterinarian can tell you. Yeah, this is the one which it tried to. Yeah, so the main is the trachea is blocked, and therefore the respiratory signs are there. So it's very, very hard to actually breathe, and then likely die. So the question the veterinarian asked, or researcher asked, that how much impact of H9 to, let's say, the vaccinated flocks. So if unvaccinated, you may have seen that up to 70 is not necessary, it is 70, that's a worst case scenario. But in vaccinated flock, largely the people are saying up to 30% reduction. And here there were 3 flocks, one flock showing more than 30% reduction, and that reduction never reached to the peak. The other maybe 20%, and then try to reach the peak, and the other is around 10%. So the question is, why 3 different flocks infected, vaccinated, and then natural infection with H9N2. Why these? Some are 30, some are 20, some are 10. The reason could be anything. One thing is it could be the delivery of vaccine is not accurate in one form than the other. The second is the challenge of virus in three different forms may be different. And of course, the vaccine efficacy is not used. The same vaccine in three different ones, their efficacy may be changed. So there is a post-vaccination monitoring will help you to determine what happened if there is a reduction and is there any co-infection or a single, in this case it is single H9N2, but there may be a much more worse if there is a co-infection. So the challenges again is that because in some countries like China and other countries, there is multiple subtypes like H5, H7, H9, H6, all are infecting poultry in some countries. The virus is continuously evolving and changing, so it overcomes the vaccine efficacy. The co-circulation of these multiple variants, so the vaccine you give is maybe only against one variant, but not one virus, but within that one there is other variants which are happy that even promote it when you give the vaccine. So the vaccine is not working against all variants. And then the main then happen is the poor quality vaccine. That means that poor is not, they make it poor, it is not antigenically matched to the field virus, and therefore the efficacy is reduced, and that you can say the poor quality. Then the other problem currently with live and inactivated vaccine is the maternal drive antibody interference, and that also directly impacts to the induction of immune response because there is a several mechanisms, but simple mechanism is your antigen, your epitope is masked with antibody, and therefore is not exposed for induction of immunity. And then, of course, the other viral infection or bacteria which reduce the immune system or the immune compromise, and therefore the vaccine unable to work. So there are not a single challenge, there are multiple challenges, so you have to tackle all multiple challenges to improve your production. So the one of the main challenge which we are looking that how these viruses evolve, as you can see a few years back in 2004 or 3, in person H7 and 3 was circulating, H5 and 1 was, and H9 and 2. Previously, when the virus came in 1998 or 99, it was a different virus. It was not causing much disease to the poultry. So it was alive, so people are not very worried about the virus. The virus was H9, but not worried. This virus then gained a gene from H7 and 3 virus and one gene from H5, and it became a perfect virus to in fact cause disease and very efficiently spread with low dose. That means you have a little virus, less than 100 particles still can infect and transmit. So that virus becomes so big, it causes disease, and then of course the farmer complaining that they are losing their production, and that's why they started the vaccination. And since then the virus, we call it genetic drift. They are changing the HA and other genes, and that gene compromised because they escaped from the vaccine. And the simple example is only in 2022, H5N1 in UK was infecting. We were sequencing, and as you can see here, more than 22 genotypes of H5N1 was isolated in the UK in 2022. Either it was in the wild bird and poultry, and we were culling all the infected bird. Still, the virus was. So the changes in the virus change the virus ability to transmit. In fact, and increase the virulence, and of course increase its ability to cross species barrier to infect mammals as well as humans. So again, when we look at here H9N2, we call it H9N2, but it is very variant. For example, here in Pakistan, the virus is similar to Iran and India, but also there is other viruses which are again in Iran, which are very distinct from these viruses. So very little data we have, which is from Pakistan. I can compare whether these viruses are in Pakistan as well. So the limited data always give some problems. Similarly, when we closely look, there is a multiple variants which are co-circulating. So you need a vaccine which can actually attack the variants, not a single type. Example, I would say, you lock it. So you lock it. Okay. So this is a very typical example. In Bangladesh, we have a project. We sequenced more than 240 isolate in 2022, and we saw that there is so many variants within the region. For example, there are three distinct clusters co-circulating in some region, and some region is a single. This cluster has these mutations. This cluster has these mutations, and this cluster has these. All these are co-circulating in different regions. Some have it all, and some are isolated or a single one. So if you have a vaccine against this one, it may not work against the variant. So you have to find out. You cannot make 240 vaccines, and you need a single vaccine to actually cover all these variants which you are saying. And for that one, you need to do the analysis, antigenicity analysis of all these. So what we have done is we made all variants of these viruses in the laboratory because using reverse genetic, and then analyze the antigenicity and then identify which strain is cross-reactive against all variants, and that will work much better than just selecting randomly any strain. And similarly, when we sequence the internal gene, we see that these are the 3 or 4 independent viruses have a different genetic makeup, and because of the internal gene, the virus ability to cause pathogenicity or virulence is also different. So the same virus H9N2 is not like it causes this typical disease. Some viruses may not cause disease, some causes are more virulent, so you have to find out. And similarly, the viruses which are in China, we call it Beijing-like lineage, or different than a G1-like lineage again, but in some region, these both are co-circulating. So that also increase the divergence and antigenic difference between these viruses. As I can see here, I should take one Vietnam virus, one Pakistan virus, and one old Pakistan virus. So the old Pakistan virus was not causing any mortality in embryo up to 72 hours. The new, which is currently circulating or isolated 2008, since it is almost same in terms of virulence, it was causing about 30–40% mortality in embryo at 72 hours, with this Vietnam virus Beijing like genotypes 57 was almost causing 90% mortality in embryo by 70. So all H9N2 by virulence is not the same. They are also different depending on its genetic makeup. Similarly, when we infected chicken and we take the isolate virus from different tissues, so the majority of the virus you can find into the nasal turbinate, but then the Pakistan virus and Bangladesh virus mainly we can say in the brain and the virus was really causing a trimer kind of convulsion to the birds, whereas the Vietnam virus was more virulent and it went into the lungs and kidney, whereas this was not, and therefore the bird was dying. But the bird was dying both cases, but it is the virus dissemination in the tissues of the infected bird is different, and that's why disease appearance is also maybe different. If you can see in one bird, it is showing different signs compared to, it depends on the strain and the variant, so it is in deep into it. Similarly, why this virus is more virulent, so we found that even a single gene exchange, in this case M or NS, can increase the virulence of a less virulent virus to a more virulent virus. And this is also we compare, for example, we take a one Morocco virus that time which was causing disease with the Beijing-like viruses, Chinese viruses, we see that Chinese viruses are causing more clinical disease in terms of conjunctivitis and clinical signs compared to the G1, but this is a single strain or two strain, but if you take a multiple strain from the same region, they may show a difference as well. Now the antigenicity. When we take the antigenicity for vaccine kind of comparison or which vaccine can work or what is the antigenic difference between different strains or different countries. So we made a lot of viruses, as you can see here, and raise the anti-sera against all these viruses. And then for each virus, we check what's. So for example, here the Pakistan viruses that time are highly cross-reactive homologous, and some of the homologous against India and Egypt. But for example, the virus in UAE, virus in Bangladesh, they are very distinct antigenically. So the same vaccine cannot be universally, can be used until you check it whether, for example, this vaccine, Pakistan vaccine, can be used in India or in Israel or in Egypt. But if you see in some of the Bangladesh or these viruses, it is not. So there is a huge antigenic diversity in the strain which are circulating or co-circulating in the same region. When we see a more closely, for example, here we have a Pakistan virus and another Bangladesh virus antigenically. When we use the Pakistan strain as anti-sera, it is cross-reactive, but not with the Vietnam virus. And when we use the Bangladesh, then it is not cross-reactive against Pakistan and Vietnam. And then when we use the Vietnam, it's only cross-reactive with Vietnam, not with Bangladesh or Pakistan. Again, this is a very kind of convincing evidence that the viruses which are circulating in the field are antigenically heterologous, not homologous. But other thing is, some people try that if it's a 2% or 4% genetic homology, that may be a similar. This is not the same. Genetic homology is always different than the antigenic homology. Example is this virus. Consider as 100% in terms of because we raised these monoclonal antibody against this virus. But this virus, again, the same time isolated, it have only one mutation which impacts on these two antibodies. But overall, as a polyclonal sera, it is the same. This virus, again, 96%, that 4% different, like this virus, completely do not cross-react at all. So antigenic homology is very distant, but genetic homology is only 4%. So genetic homology do not always is the similar to antigenic homology. Similarly, a few years back, we isolated viruses from Pakistan and analyzed genetically and antigenically. There were so many mutations, but only one mutation, which is the co-circulating viruses, at position 180. Instead of in some viruses at A, some viruses at B or B, and that is causing a significant antigenic change. So if you have even 400 viruses or 400 mutations, only it could be a single mutation in variant can compromise the vaccine efficacy. And this is example from 2008 when we see these positions are changing in terms of percentage at both position and 180 and 183 in most of the world viruses, either it is in China or in Pakistan or in Egypt. And if, for example, if you have a vaccine against the virus has A, it is a very high cross reactivity, but if the T and V, it lost more than 90%. And similarly, if you have a N, you raise the anti-C that do not cross react. At the same position, you have a D. So the variant, the problem is that poor circulation of variants may compromise the vaccine itself may be good for the homologous, but you need to know what are the variants and whether these variants are effective or your vaccine is effective against these ones. So in summary, H9N2 antigenically variants are co-infecting poultry and have ability to overcome the vaccine effectiveness. As last almost 20 years, we identify several mutations in HA, H9HA, and some of it which are showing red cause more severe antigenic distance or diversity compared to other. Therefore, there is a need a continuous surveillance of variants of circulating in the field and then antigenically match the vaccine that can offer a more broader protection against the variant and reduce the impact. Again, control strategy. The first thing is, again, you have to risk assess. What is the risk to your poultry? Is it, for example, I gave at 7 and 9 in China, in Chamar, it was not causing any disease to poultry, but it was highly infectious and fatal to humans. So the farmer do not want to control it because it is not causing any disease to poultry. So what is the risk of it? Is it a risk to poultry? It is a risk to animals or humans? And that risk basis and whether this is virulent, how much virulent, like low path and high path and so on. And on that basis, if you are adopted one vaccine and then what vaccine you are using against your field viruses. And most of country here, Asia, they adopted vaccination, and here is most of the country in Europe, they adopted that they die and come. So that is a clear difference. So what my laboratory did is we developed a pipeline because we do not get the viruses from all over the world, but we can get a sequences from the world. Because a lot of viruses are being sequenced, put into the database, so we retrieve the sequence, and then we do bioinformatic analysis to see which are the dominant one, which are the variant one, and then we make the field copy of the virus by reverse genetic, and then check its replication pathogenicity in vitro and then vivo, and then do a different assays such as receptor binding, pH fusion, thermostability to check is it stable, how much it is a risk to human, or then its antigenicity in terms of vaccine, whether this escape from the vaccine or the current antiviral are available for a human. So we check it whether it is resistant to that virus or not. And then study its markers or what the mechanism of evolution and then this prediction, and then develop the vaccines and diagnostic. And this is example, the current H5, and one virus emerged in UK, and some of the viruses had a single mutation which increased the ability of the virus to bind to human receptors, so it increased the zoonotic potential. At the same time, this mutation antigenically so much distinct that if this virus circulates, the vaccine against this virus will not work. So by doing reverse genetic, first we can do the bioinformatic to select a more cross-active antigen, and then we do the reverse genetic to make the virus in the laboratory, and then we can engineer antigenic epitope to increase its broader cross-reactivity or antigenicity, and then we can manipulate the vector like NDV, HDT, and so on to clone these genes into the vector to make the vaccine. And then we are also making passive immunization. We in vitro can express the antibody very cheaply, and then you can actually spray it by cloud or get in the water, so you can protect that way to the bird. So that is our recent experiment show. But I do not have much time to go through all, but I will try a little bit. For example, Bangladesh, these are so much viruses. We follow this technique of which I described earlier and generated the viruses by reverse genetic, selected the viruses, raised the anti-sera, and from them we see that the one of this virus which we selected is more cross-reactive compared to if we select the viruses from here or there. And similarly, the viruses which are I described earlier at position 180, or this virus, Pakistan virus, antigenically homologous, but these variants are also circulating in Pakistan. They are more than half the antigenic compared to this. That means these variants, if you give the vaccine, may continue to circulate and you are giving vaccine. So you need to select a vaccine. So if we selected this virus which has a T instead of A, and that induces immunity against all variants equally rather than this disparity. So selection of antigen is very critical for your vaccine development and delivery. Similarly, we manipulate the antigen. For example, here we manipulated the glycosylation on the HA. In this sense, we added a glycosylation at that site, and that increased the cross-reactivity and immunity significantly, as you can see, 167 to 2000. Similarly, we have modified the neuraminidase in a way that it induced more immunity against the different variants. So in that sense, we are selecting these vaccines. We also selected antigen, for example, here H5N1. So here the previous H5N8 do not induce immunity against the current H5, whereas if we use a selected H5, this is a more cross-reactive against H5N8 and other compared to using the old H5. Similarly, here we modify the antigen by the antibody to the antigen which deliver or take the antigen to the antigen presenting cell receptor, and that induce immunity. And that way we see that in the bird which has very high immunity, the commercial vaccine failed to induce immunity, whereas here, even up to 90 days, the immunity is more than 10 HI titer. That means that changing or delivering directly antigen to antigen presenting cell induce a very high immunity. Similarly, this high immunity can overcome so many variants we have, which was not cross-reacting with the normal vaccine, but this target vaccine induced a very high immunity against the variant. We also use recombinant Newcastle disease virus, and we put Newcastle disease virus, we put the HA of H5, H9, or other. Muhammad Munir may give a more detail, but here what we do is, in this case, H9HA we put, and we saw that by using this scaled vaccine, ND, we can protect against the ND as well as the H9. and that means you can reduce the egg production up to 50% for the same vaccine you are currently using. Similarly, we manipulate the herpes virus of turkey. We can express Newcastle disease or H9 or H5, and then giving to the chicken, we saw that we can induce immunity, very high immunity, even on 14 days or 21 days against both H9 as well as ND, and of course the marriage disease. We also produce engineer antibody, or we say chickenized antibody, and then we give in the chicken by experimentally we give a 1 drop intranasally or ocular, but in the field you can do spray or give in water. And then we saw that here the control bird have a huge amount of virus, where the treated bird have very little or negligible virus. So the treated bird do not show any clinical sign, and we give a very high dose of H9N2, and certainly the treated bird in the contact also didn't show any transmission. So in summary, continuously AIV variants are continuously emerging, posing risk to poultry, livestock, wildlife, as well as the human. Selecting cross-reactive antigen can provide more protective immunity against variants, and certainly implementing innovative approaches which we are developing can help to reduce the impact of these viruses in poultry and certainly transmission to humans. And this is currently my group at Poultry, and these are the previous PhD and postdoc which was contributed in different areas of my research or my group research. And thank you very much for listening.